write anything down. This diagram will not be on the test. I just wanted to talk a little bit about this because I think it kind of helps you understand how the brain has these cavities inside that are called the ventricles. And that's actually what we're going to talk about first when we get to the brain in just a little while. So this is actually how the central nervous system begins. It begins as a tube. It's a hollow tube called the neural tube. This starts forming in an embryo in the 3rd week of gestation. Okay, so the neural tube goes from the head end to what's called the tail end of the embryo. And the head end will start developing these 3 swellings. This is what's going to eventually become the brain. At this stage, this is just referred to as the forebrain, midbrain, and hindbrain. You may have heard those terms before. The rest of the tube becomes the spinal cord. Okay, so eventually these 3 swellings will become 5 swellings that you see here. And these 5 swellings of tissue, and if you're wondering like, well, where is the brain itself? Actually what happens is, as it develops, the wall here gets thicker and thicker and thicker, forming the structures of the brain. Okay, so these 5 swellings will go on to form all the structures of the mature brain, which are listed here. So for instance, the Mickey Mouse ears, what looks like Mickey Mouse ears at this stage, will become the cerebral hemispheres, okay? So again, what I wanted to show you is just notice again, there's a lumen inside. The lumen here actually remains as the ventricles of the brain. So this table is kind of handy because it actually tells you what ventricles are in what part of the brain. And we're going to be talking about that in just a little while, okay? So I just think it's kind of helpful to see, oh yeah, okay, so there's a lumen and the brain forms around it. And we end up with these spaces inside. Same thing with the spinal cord. We end up with this narrow canal that runs the length of the spinal cord. It's called the central canal. Okay, so again, you don't have to know this, just a little FYI. Okay, so let's talk a little bit about how neurons are organized, and then we'll start talking about the brain. So this is stuff we kind of mentioned last time. I said that whether you're looking at the central nervous system or the peripheral, neurons are organized, which is what we would expect, right? You wouldn't expect neurons to just be scattered randomly anywhere. They have to be organized so that the neuron cell bodies are grouped together and then the axons travel together in an organized way. And that way they can send action potentials from one area to another. Okay, so just some terminology for now. In the central nervous system, we call a grouping of neuron cell bodies a nucleus. Okay, so you don't want to get this confused with the nucleus of a single cell. This is different. This is a little cluster or grouping of neuron cell bodies. And again, this is just how neurons are organized. I'll show you what this would look like in just a little while. We'll look at a diagram. Okay. When we start talking about the brain, we will talk about some of the nuclei in the brain, and we'll talk about their function. And so what that means is that when a group of neurons are located together in a little grouping, they will have the same function. Okay? And as you guys know by now, these specific nuclei have specific names, right? Everything has a name. So this is a general term. Okay? Okay, so we know that in the peripheral nervous system, we have axons that travel together in nerves, right? Yes, we do know that. Okay. So in the CNS, do we have nerves in the CNS? No, we do not. But we have something similar. We have groups of axons traveling together, but they are called tracts. Okay, so this is similar. Tracts do not have those layers of connective tissue that the nerves have. But it's a group of axons going from point A to point B. Okay, so depending on the direction that the axons are traveling, okay, we have different types of tracts. Okay, so let's talk about these. First of all, if the axons cross over the midline connecting the right and left sides of the brain or spinal cord, it's called a commissural tract. Okay? So these cross over the midline. That is your key word here. They cross the midline, connecting the right and left sides. The largest commissural tract in the nervous system is the corpus callosum. Okay, corpus callosum. This supposedly means tough body. Tough body. I don't know if that name doesn't really help you much. I think of it as being more of a communication, pathway for connecting all the different parts of the right and left sides of the brain, but that's what the name means. Okay, and we will see this in just about every view of the brain that you look at. You will see the corpus callosum because it's big. So we'll take a look at it in just a minute. Okay, if the axons connect different parts of the same side, like they do not cross the midline, it's called an association tract. Okay, so these do not cross the midline. These could be very short. They might go from one little gyrus to the next, or they could be longer where they go from, say, like the frontal lobe back to the occipital lobe, but they connect structures on the same side. Okay, and then finally, projection tracts. Projection tracts are where axons travel vertically. And you can imagine we're going to have a bunch of these between the brain and the spinal cord because they have to communicate with each other. Okay, so if the information is traveling up to the brain, that is called an ascending projection tract. Okay, ascending. The information is going up. What type of information do you think that would be? Sensory or motor?
Sensory.
It's going to be sensory. And then we also have descending projection tracts where the information is going down or action potentials are going down. That would be?
Motor.
Motor, right. Okay, so projection tracts can be ascending or descending. Okay, we'll take a look at a diagram that shows these types of tracts in just a few minutes. Okay, so this is the terminology for the CNS, okay? Of course, they have to come up with different names for the PNS, right? Has to be different. Just to make it harder for you. Okay, you guys good with this? Any questions? Okay, so in the peripheral nervous system, same thing, our neurons are organized, but here a group of neuron cell bodies is called a ganglion. Ganglion is singular, ganglia is plural, just so you know, okay? I think we actually mentioned this term last time. Okay, and then you guys know something about nerves. We looked at the structure of a nerve, and a nerve is a group of axons traveling together in the PNS, right? We talked about how we have two groups of nerves, cranial nerves or spinal nerves. We talked about those layers of connective tissue, but this is the definition of a nerve. Okay. Okay, and then we also talked a little bit about white matter. White matter is formed by what? What is that substance called? Myelin. Okay, let's talk a little bit more about white matter and gray matter. Okay, so we said that myelinated axons make up our white matter because myelin is really what forms the white matter. So what is gray matter? Well, gray matter is everything else. It's going to be our neuron cell bodies and also dendrites. These are not myelinated, okay? These are never myelinated. And we also do have some unmyelinated axons. So all of this stuff makes up gray matter. Okay, and then again, the white matter would be all of our myelinated axons. We'll take a look at some sections through the brain and the spinal cord, and we'll see that white matter and gray matter are kind of separated, which again, it kind of makes sense, right? Because we talked about how we have groups of neuron cell bodies located in distinct, clusters, and then we have areas where the axons travel together. So they're kind of separated. You guys have other questions? Any questions? Okay, so this is a table that goes through some of these terms, gives you a definition. Let's take a look at some white matter and gray matter. This is a frontal cut through the cerebrum, right? This part of the brain up here. And here we have a little bit lower down in the brainstem, a couple of other sections. This is a cut through the spinal cord, okay? So whatever part of the brain or spinal cord that we're looking at, again, notice how white matter and gray matter is kind of separated into different areas. Okay, so taking a look at this section through the brain, notice that around the outer edges of the brain surface, we have this gray matter. Okay, so gray matter isn't literally gray, it looks kind of like a darker tan color. And so this part of the gray matter actually is very important. This is where most of our neuron cell bodies in the entire nervous system are located. This is the area that is called the cerebral cortex. Okay, cortex meaning it's the outer edges. There are about 6 layers of neuron cell bodies here, but this amounts to billions and billions of neurons, billions and billions of neuron cell bodies. Anything that we're aware of on a conscious level is happening somewhere in the cortex. Okay, so of course, all these other areas of the brain have their own functions, obviously very important because they all have to work together, but the cortex is where we're able to think, consciously. So you're hearing the sound of my voice, you're understanding the words, you're thinking about your day, whatever you're doing is happening somewhere in the cortex. And we'll come back to that. Okay, and then these other areas, notice here we have some gray matter. These would be nuclei. These would be groupings of neuron cell bodies. Okay, again, they're going to have their own specific names, but they are nuclei. This tract, remember a tract is a group of axons traveling together. So all of this white matter, this would be myelinated axons. There might be some unmyelinated mixed in, but it's mostly myelinated, which is why it looks like white matter. So notice there's a tract that goes across the midline. So this tract here would be the commissural fibers or axons that we talked about, connecting the right and left sides of the brain. Right? Remember, commissural goes across the midline. So this would allow the 2 sides of the brain to communicate with each other. This actually happens to be the one that's the largest. This is the corpus callosum, okay? The largest commissural tract in the nervous system. We'll talk more about that later. Okay, and notice that we see some spaces here and there. There's another one here. These spaces are the ventricles, and we're going to talk about those next, okay? So I think that's about it that I wanted to show you on these diagrams. Oh, and there's one more diagram that shows the different tracks that we talked about, depending on the direction that the fibers are traveling. And axons are often called fibers. It's the same thing, okay? Axons, you can say axons, fibers, and tract. These words are often used interchangeably, okay? Okay, so notice here, this is just showing one side of the brain, just one half. So all of these different axons these are examples of association tracks, right? They don't cross over the midline. Again, these can be very short, or they can be longer, but as long as they don't cross over the midline, it's an association tract. Okay, now notice in this key here, they're showing you a couple of different names for these. Don't worry about these names. They are association tracks, okay? They're all association tracks here, meaning they don't cross over the midline. Okay, now here we see, notice the green axons are traveling vertically, connecting the brain to the spinal cord. So these would be projection tracks, right? They could be ascending or descending. You can't tell in this picture which way the action potentials are traveling. These are projection tracks though. Okay, and then this one again is the corpus callosum. It is a commissural tract, but it happens to be the largest one, which is the corpus callosum. Okay, so these diagrams just show you the different types of tracks depending on the direction that the axons are traveling. Okay, that's all this shows you. And I think I gave you guys this figure number. These could be on your practical exam. I might ask you what type of tract is shown. Okay? Any questions on any of that? Okay. So, before before we start talking about the ventricles, let's just run through the parts of the brain, like the regions of the brain, so we know what we're talking about when we start getting into the brain. Okay, so notice here in this diagram, you're looking at the lateral view, and this big thing here is the cerebrum. This is referred to as the cerebrum, or sometimes called cerebral hemispheres. There's a right and a left cerebral hemisphere. This is very large in humans, very developed in humans. You'll see when we look at the sheep brain next time, of course not as large in sheep. And it kind of comes around part of the brain stem, so you can't see some of the parts of the brain that are kind of up underneath this, okay? And this is divided up into lobes. You guys already know the names of the lobes because they're named like the bones of the skull. So these would be frontal, up here would be parietal, back here, occipital, and temporal, okay? Let's take a quick look here. I just want to show you, just as kind of a little introduction to a bunch of parts of the brain. I'll try to hold these together. Okay, so in the big brain I have here, notice these little crevices that are all over the place, and you guys know when we have folds, what do the folds do for that tissue? Increase surface area, right. We said that the outer few layers of, the outer few millimeters of tissue here is the cerebral cortex, where we have all those neuron cell bodies. So this increases the surface area for more neuron cell bodies. These little hills are called gyri. Oh, got it. I was gonna need some help there. Okay, so these little hills, each one of these is a gyrus, or plural is gyri. That's a Y, gyri. And these little crevices here that you see on the surface, each of these is a sulcus. And plural, technically plural is sulci. You pronounce this word sulci. Some people say sulci. I don't really care. Sulci, sulci, whatever. Okay. The deeper crevices, like this one here, okay, this one here is actually very deep. This is a fissure, okay? So there are two fissures I would like you to know. This one is called the transverse fissure because it is in the transverse plane. So again, a fissure is like a deep sulcus, okay? It's between the cerebrum and the cerebellum. Okay, and then the other fissure is this one. Okay, this is called the longitudinal fissure. It's between the two hemispheres. I don't know why it's not just called sagittal, but it's called longitudinal. Okay, this one here. So we have the longitudinal fissure and we have the transverse. I don't think that your book labels this anywhere in any diagram. It mentions it in the text, but it doesn't label it. And I think it's because they forgot. I think they forgot the label. Mm-hmm.
Do you mind repeating the difference between gyri and association?
Yes. Okay. So a gyrus is the little, the raised, like the hill. Okay. The sulci are the cracks or crevices between them.
Okay.
Does that make sense?
Yeah.
Okay. Okay, so let's take a look at a mid-sagittal cut so that we can see the other parts of the brain. Oh, actually, first, this is an inferior view. And on this inferior view, you can see all of these cranial nerves because they actually exit from the inferior or kind of more anterior part of the brain stem, the brain and brain stem. So that's what you see here. These will go out to mostly tissues that are within like the head area. Okay, like you can see the optic nerves come from the eyeball. These carry visual images. These are the olfactory nerves that would carry smell. Right. Okay, so this is a mid-sagittal cut. And just so you guys know, those figure numbers that I gave you, if they show you a diagram and then a photograph of an actual brain next to it, the photographs will not be on the test. I'll use the diagrams. okay? Just so you know. Okay, so here we have this mid-sagittal cut. Notice again, this is the cerebrum, this guy here. Okay, very large. This is that transverse fissure that we mentioned before between the cerebrum and cerebellum. Okay, this is that corpus callosum, that really large commissural tract. So these axons would be coming out toward you. If you could see them, you would be looking at like the cut ends of the axons here, because remember they cross over the midline connecting the right and left sides. Okay, so this part of the brain right here, I'm going to outline it. Notice it's right kind of under the cerebrum. The cerebrum sits on top of it. This is called the diencephalon. Long name. Okay. The three parts of the brain stem, or the three regions that are inferior to the diencephalon, make up the brain stem. Okay, so they would include this region here, about an inch long. This is called, you can say mesencephalon. I usually just call it the midbrain, but you can also say mesencephalon, which means midbrain. Okay. This next area, notice it's like this big anterior bulge here. This is called the pons. And then the last part of the brain stem, this kind of, again, not very long, a little bit of an anterior bulge here also. Below the pons, this is the medulla oblongata, usually just called the medulla. So these three regions, the midbrain, the pons, and the medulla, together they make up the brain stem. Okay, so notice that here we have the cerebellum. Okay, the cerebellum looks like kind of like a miniature cerebrum. Actually, cerebellum means little brain. This is a really important area for motor coordination and balance. It is not part of the brainstem. It's posterior to the brainstem. Okay, it's a separate region of the brain all on its own. Okay, cerebellum. Okay, so those are the 1, 2, 3, 4, 5, 6 major parts of the brain. And the medulla, one other thing I wanted to mention is it's continuous with the spinal cord. It actually, the boundary here is actually the foramen magnum. So when it goes through the foramen magnum, then we call it the spinal cord. Okie dokie. Questions? Okay, so the first thing I want to talk about are the ventricles. And again, these are spaces inside the brain. What's inside the ventricles? What are they filled with? Cerebrospinal fluid. Okay, so last time when we talked about the different neuroglial cells, you guys remember the different neuroglial cells in the CNS, right? Of course you do. Okay, so which are the ones that line the ventricles? Starts with an E. Did you say it? Ependymal. Yes, very good. Ependymal cells line the ventricles. Remember, this is the neuroglial cell that's really a type of epithelium. It's simple cuboidal with cilia. Okay, so this lines the ventricles and also forms cerebrospinal fluid. I'm just going to abbreviate cerebrospinal fluid, okay, CSF, in a structure called the choroid plexus. Okay, so what is the choroid plexus? Actually, this is very simple. The choroid plexus is in the wall of each ventricle. Each ventricle has an area that has a choroid plexus. This is just the ependymal cells that line the ventricle and the underlying blood capillaries. Capillaries are the tiniest blood vessels in the body. They're microscopic. So this is ependymal cells, okay, and the underlying capillaries. So what happens here is some of the blood plasma in these capillaries will leak out across their wall. They'll go through those ependymal cells, and they'll be in the cavity of the ventricle. Okay, that plasma will be in the cavity of the ventricle. Once that fluid is in the ventricle, we call it cerebrospinal fluid. Okay, so I will show you this on a diagram. I think it will make more sense when you can kind of see a diagram. But cerebrospinal fluid is very similar to blood plasma. They're almost the same in composition, and so it makes sense because CSF comes from the blood. It's going to circulate through the ventricles, and it will eventually get reabsorbed back into the blood. And we'll trace that pathway. Okay, so this will kind of give us an introduction to a bunch of parts of the brain as we go through this. Okay, so notice here on this mid-sagittal cut, you don't really see the ventricles very well, right? Actually, you don't really see them here because they're kind of inside the tissue of the brain. You can see a little bit of this fourth ventricle, which is right here, but you don't really see them well in that type of diagram. Okay, so this is what we're going to talk about. This lists the ventricles for you, but I want to show them to you on this diagram. So this is where you can see them the best. This is what is called a phantom view, like you're looking through the tissue of the brain, and you can see the shape and the position of the ventricles, okay? It looks just like this model. So we have this model of the ventricles, okay? Da-da-da-da. So notice this looks just like this, exactly the same. Okay, so you can see the ventricles here, you can see them on this diagram. This actually was made from a real human brain where they put this material, whatever this stuff is, some kind of plastic, into the ventricles and it hardens. And so they have an exact mold of the ventricles from a real human brain. Okay, so let's talk about these. And as we go through these, I would like you guys to know what part of the brain they're located in, okay? So first of all, notice these ventricles here, side by side. These are located in the cerebrum, okay, up in the cerebral hemispheres. These are called the lateral ventricles, just right and left lateral ventricles, okay? Because they are the largest, they will make about 80% of the total volume of CSF. Okay, so we said that every ventricle has a choroid plexus, right? The choroid plexus would be located kind of here in the floor of the lateral ventricles, and that makes about 80% of the total volume of CSF. So cerebrospinal fluid will go from those blood capillaries, remember, from the blood capillaries will go into the ventricles. It will now go from the lateral ventricles, it's going to go down into the next ventricle. So CSF is going to kind of circulate through these spaces. Notice there's a little connection right here from each lateral ventricle into the next ventricle. These little connections are called the interventricular foramina. Mm-hmm.
You said that the right and left lateral ventricles make up 80% of what?
CSF.
Oh, okay.
Yeah. So these little connections right here that allow CSF to go from the lateral down into the third ventricle are called the interventricular foramina. So on this model, when you guys are looking at this model, it looks like this thing here is a big hole, right? There's actually what looks like a hole here in the third ventricle. Notice that it looks like there's a hole. Okay, when you're looking at this though, what you have to try to remember is this whole thing is like a big hole, right? These are spaces. So what actually passes through here is a solid piece of tissue connecting the two sides of the brain. Okay, we'll talk more about that later. So these interventricular foramina, when you look at the model, don't look for a foramen. You want to look for the connection between the lateral and the third ventricles, and you'll see this when you look at it. There's like a little connector right here, okay? A little piece of what would be tissue, okay? Or what would be a connection, a foramen in the brain. Looks like a little piece of plastic on the model. That is the interventricular foramen, okay? Okay, so CSF will go through those interventricular foramina into the next ventricle. This is called the third ventricle. This is right in the midline. There's only one of these. It's right in the midline. This is in the area called the diencephalon. Okay, so diencephalon. That part of the brain below the cerebrum. And I'm sorry, let me do this. Sorry, you guys. I should label that as third ventricle. So this is the third ventricle, and it's in the diencephalon. That's better. Okay. From here, CSF is going to go through this really narrow channel. This is called the cerebral aqueduct. Okay, this goes through the midbrain, travels through the midbrain. Okay, and it connects the third to the fourth ventricle. Okay, so the fourth ventricle is here, it's the smallest. This is actually in the area of the pons and medulla. Okay, so pons and medulla. Okay, fourth ventricle. Okay. Mm-hmm. No. So when you're identifying, if I were to ask you to name the ventricle itself, you would want to say fourth ventricle. If I ask the area it's located in, yeah, you want to be specific. So on this, for this unit, what you want to think about is, we'll talk about a bunch of different structures that are in these different, kind of bigger regions of the brain. So you guys always want to be as specific as you can and name the exact structure or the exact part of the brain. If you're more general, you would probably get a half point. So for brainstem, I'd give you a half point for that. Okay? Okay, so this is where cerebrospinal fluid is now going to leave the ventricles. It's actually going to go out of the fourth ventricle, and notice it has these openings that are called apertures. I think the reason these are not called foramina is so you don't get them mixed up with this one here. Okay, so these are called apertures. There are two lateral apertures and one median aperture. And it's kind of hard to see them, but you can kind of see where there's little openings, okay, that are opening out of the fourth ventricle. This allows cerebrospinal fluid to go out into a space where it will now circulate around the brain and around the spinal cord. Okay, and we'll talk about that in just a few minutes. Okay, so you guys have questions on this? One thing I just remembered, I forgot to tell you that looking again here at the lateral ventricles, you can see how they have these weird projections that stick out. These are called horns, okay? Actually, very simple. These are called the anterior horns. The anterior horns are in the frontal lobes of the brain. Okay, these are the posterior horns. These are back in the occipital lobes. And then these inferior horns are in the temporal lobes. Okay, so I'd like you to be able to identify the horns. Those are easy. Alrighty, questions? Okay, so when cerebrospinal fluid leaves the fourth ventricle, it's going to now go into a space called the subarachnoid space. And to understand where that space is at, we have to talk about the meninges, the membranes that cover the brain and spinal cord. Okay, so let's talk about meninges. Oh, and this is just a diagram that shows you the choroid plexus. Okay, so again, the choroid plexus is just the ependymal cells that line the ventricles. And again, you can see this is like a simple cuboidal epithelium with cilia and the underlying capillaries. So as blood plasma just kind of leaks out, it will just kind of slowly leak out across the wall of the capillary. It's going to go across those ependymal cells, and now it's out in the ventricle. Or this could be the central canal of the spinal cord, either one. Okay, and so it leaks out into that space, and we call it cerebrospinal fluid. Okay? And this is showing a cut through an actual brain. This is what the choroid plexus would really look like. I know it's kind of hard to see what that really is, but this is part of the lateral ventricle. It's one of those horns, and this is it's just blood vessels kind of covered with the ependymal cells. Okay, so let's talk about meninges. Okay, so you guys have heard of meninges, right? Because I know you've all heard of meningitis.
Mm-hmm.
Yes, something we do not want. Okay, so there are 3 membranes that cover the brain, and these also surround the spinal cord. These are basically the same around the brain and the cord, basically the same. The one layer that is different is this one, the dura mater, okay? So around the brain, the dura mater has 2 layers. So it's a little bit more involved around the brain. Around the spinal cord, it's just 1 layer. Okay? Dura mater literally means tough mother. And it is very tough, fibrous connective tissue. It's dense, irregular. Okay, so again, just to remind you, it's 2 layers around the brain, 1 layer around the cord. So we're just going to talk about this around the brain for now, since we're focusing on the brain. Okay, so the 2 layers of the dura mater around the brain are called the periosteal and the meningeal. Okay, so the outer layer is right up against the periosteum of the skull, right? You guys know periosteum is the membrane that covers bone. So it's right up against the skull, so it's called the periosteal layer. The deeper layer is called the meningeal layer. Okay, so this meningeal layer of the dura mater is going to come into the fissures that we just talked about to kind of wrap around and protect the parts of the brain, okay? Where it comes down into the fissures, it has special names. Okay, so let's talk about these. Let me get my dura mater guy. Okay, so this is Mr. Dura mater. And notice this model does not have a brain. Okay, it's supposed to be that way because this is just showing the parts of the dura mater. So that is what this tissue is here. Okay, so remember we said there's a longitudinal fissure here, right? So this piece of dura mater that comes down into the longitudinal fissure between the cerebral hemispheres, this is called the falx cerebri. Okay, falx means it's C-shaped. That's what that word means. Okay, so falx cerebri is between the cerebral hemispheres, right, in the longitudinal fissure. Okay, then we have another piece of the dura mater that comes into that transverse fissure between the cerebrum and cerebellum. That is called the tentorium cerebelli. Tentorium cerebelli. So this is in the transverse fissure. Okay, so because this is like in the transverse plane, it's like a tent, right? It's like a tent between the cerebrum and the cerebellum. Okay, and then finally there's a falx cerebelli. This is a really short little piece of the dura mater between the 2 cerebellar hemispheres. So when you look at this model, you will see these pieces of dura mater. Again, this one here is the falx cerebri. This one is going to be the tentorium cerebelli. Okay. And then this one here on the base, you actually have to look at the base of the model, is the falx cerebelli. Okay, very short. Okay, so that is what this model is for. Okay, one more thing about the dura mater that I would like you to know So we have these two layers of the dura mater. Where those two layers separate from each other, there are spaces that will actually fill with blood that's coming from the brain. And these spaces will collect the blood and it will eventually go through our internal jugular veins back to the heart. Okay, so let's take a look at that. Notice this diagram looks like the model. Okay, so this diagram is really just like this model. Again, falx cerebri. This whole thing right here, you're looking at like the cut edge of it. This is the tentorium cerebelli. And then with that little short falx cerebelli here. Okay, so notice you see all these things that look like blue stripes, right? These are actually filled with deoxygenated blood. On models and diagrams, deoxygenated blood is always shown in blue. In the body, it's like a dark red color, but they show it in blue just so you know it's deoxygenated, not oxygenated, okay? So this is the blood that actually is coming from the brain. It's going to go back to the heart, okay? So these are kind of like large veins. They are between the layers of the dura mater though, okay? So we call them dural venous sinuses, okay? Dural because they're between the layers of the dura mater. Venous blood is deoxygenated blood usually, and sinus means space. It's just a space between the layers of the dura mater. So these are all dural venous sinuses, all of these things here. There's a bunch of them. But there is one I would like you to know the name of for now, and that is this one right here. This one is kind of special because this is where cerebrospinal fluid gets reabsorbed back into the blood. This one is called the superior sagittal sinus. Okay, so this one not only has this deoxygenated blood coming from the brain, but it also is where CSF gets reabsorbed back into the blood. So this is one of these things, right? This is a more general term, and this is one of them, okay? It's one of the dural venous sinuses. Okay, so on the model, this is it. This blue stripe up here is the superior sagittal sinus. So on this model, there's like 4 things that you guys need to know. The superior sagittal sinus, falx cerebri, tentorium cerebelli, and then the falx cerebelli. Okay? Okay, so questions on that? Okay, so let's talk about the other 2 layers of the meninges. And we'll kind of run through how cerebrospinal fluid goes into this subarachnoid space. Okay, so this is what we just talked about, superior sagittal sinus. So the next layer of the meninges, going a little deeper, is called the arachnoid. Arachnoid means spider-like. So you can say arachnoid or arachnoid mater, either one is okay. This is a really thin, delicate tissue, and little pieces of it hang down, and it makes it look like a spider web. Okay, so something that this tissue forms is these little structures called arachnoid villi. Okay, arachnoid villi. They're like little flower-like structures. These stick up into the superior sagittal sinus. Okay, they project up into that space we were just looking at. So CSF is going to travel through these. Okay, into the superior sagittal sinus. Okay? And we'll come back to that. Okay, and then the deepest layer of the meninges is called the pia mater. This means tender mother. This is actually fused to the surface of the brain and to the surface of the spinal cord. So when you guys are looking at the sheep brains next time, you'll see this. You won't be able to remove it because it's actually fused. It won't look like anything special, but it's there. Okay? So those are the layers of the meninges. Now let's just talk about the spaces between them. Okay. This is actually another diagram showing all of the meninges. This is a frontal section. Okay, so notice this is the bone of the skull. And here you see those two layers of the dura mater. You can see how the periosteal layer stays right up against the bone, but the deeper meningeal layer, it's going to separate away from that, and it will come down into the longitudinal fissure here, forming the falx cerebri. So this is the falx cerebri here. Okay, then you can see the arachnoid is shown in pink. Here's a little arachnoid villi. And you can see how the arachnoid villi are sticking up into this space. This is the superior sagittal sinus, again shown in blue. So it's going to extend back like that. This is a frontal cut, so it looks a little different here. Okay, and then the deepest layer, notice it follows all these convolutions of the brain surface. That is the pia mater. Okay, so this actually looks kind of like this model here. You can see on this model, this is actually for the meninges and the flow of cerebrospinal fluid. There is a frontal cut here that looks just like this. Okay, so the spaces between the meninges. These are important because you guys may have heard of these before. Sometimes people hit their heads and they bleed into these spaces, and it can be fatal very quickly because there are a lot of blood vessels, especially in the epidural space, and so they can sometimes not know that they're bleeding and just don't even go get it checked out. And I'm sure you've heard of cases like that where the person dies within a short time. It's called an epidural hematoma, or you can also have a subdural hematoma. So that's why it's important if you hit your head to make sure you get checked out. Okay, so anyway, the epidural space, epi means outside of, right? So notice that these names will tell you where the space is located. Okay, if you pay attention to the name, it tells you where it's at. So the epidural space is outside of the dura mater. But remember we said around the brain, the dura mater has those two layers, and the periosteal layer is right up against the bone, but it is not fused to the bone. So there is what is called a potential space there. Okay, so again, sometimes when people hit their heads, they bleed into that space, and it becomes an actual space. A space is created as they bleed into it. Okay, so around the brain, this is what is called a potential space, meaning it becomes a space if something gets in there that's not supposed to be there. But around the spinal cord, the epidural space is an actual space, because around the cord there's only one layer of dura mater, right? There's only one layer. And I'm sure you've all heard of epidural injections. They're given a lot for childbirth. And so this is the space that the anesthesia is injected into, okay, the epidural space. That's why it's called an epidural. Okay, and so around the spinal cord, this is filled with fat. It helps to cushion the spinal cord. Also loose connective tissue. There are blood vessels and nerves that travel through that space. But this is all around the spinal cord. Okay, around the brain, there shouldn't be anything in that space. Okay, the next space is called the subdural. Notice sub, again, it tells you where it's located. Sub means deep to. So this is deep to the dura mater. Okay, and this is also a potential space, because normally the dura mater and the arachnoid are right up against each other. Okay. And then finally, the subarachnoid space. Okay, so the subarachnoid, deep to the arachnoid layer, is an actual space. This is where cerebrospinal fluid is located. So when people have a spinal tap, or they're having their cerebrospinal fluid analyzed, it is, withdrawn, with a syringe will withdraw some of the cerebrospinal fluid from this space. It has to be, the needle has to go into the subarachnoid space so they can get some of the cerebrospinal fluid from this space.
What do they need it for? What do they need it for?
It can be analyzed to help make diagnoses, you know, when people have different conditions affecting the central nervous system, or to, you know, like I said, to try to make a diagnosis, or to try to, you know, see the progress, you know, if they're being treated for a condition that's affecting the central nervous system. So different, you know, they look at the normal composition, or how the composition has changed, you know, depending on what, you know, might be whatever condition. If they have, say, meningitis or some other condition that's affecting the central nervous system, then the composition of it can change. There could be bacteria in it, or, you know, cancer cells, depends on what they're, you know, being tested for.
Why do doctors recommend that, like, college students get that meningitis vaccine?
You know, meningitis, and I don't know why this is, I actually haven't done a lot of reading on it, but meningitis seems to be very contagious. There are certain types, like viral meningitis is very contagious, and it can spread through, like, college dorms especially. So when a lot of college students are in close contact for a lot of their day and night, then it can be, you know, it can really spread rampant, you know, it can be kind of rampant. And it's, you know, it can be a really serious life-threatening condition. So I don't know that, you know, if you're not in a dorm situation, that it's that critical, but I think, you know.
But how is it contagious? Because isn't it, like,
Because it's
inflammation?
viral. It's like any other virus. I think it's probably spread by just, you know, droplets that are in the air. I'm guessing. Honestly, I don't know that much about the different types. But, and there may be bacterial types that are also contagious. I should read up on that. I don't know that much about the different types, but.
Is it similar to like a cold? How just, well, not like a cold, but I'm saying as in, that's how
Yeah,
contagious it
similar
is.
to, you know, how cold or the flu is spread.
Yeah, spread easily.
Again, I'm not sure how easily, but I know that it's contagious. And again, certain types are contagious. Yeah, sorry, I don't know more about that. Okay, so let's take a look at this diagram, just to kind of put all of this together and follow the pathway of flow of CSF. Okay, so notice again, this diagram is like this model. It's exactly the same. Okay, so if you look at this, you can see here, and actually they trace this for you here. So notice that you can kind of see, this is another one of those phantom views, like you're looking through the tissue, and you can see the choroid plexus of the lateral ventricle here. Okay, so if we start in the lateral ventricles, cerebrospinal fluid will then go down through that little interventricular foramen, and that would be right about here, where you see the arrow, okay? So it's going to come down into the third ventricle. The third ventricle would be right here. This is the diencephalon. This is literally, the third ventricle is right in the midline, like where my hand is, okay? And this is the choroid plexus of the third ventricle. So remember, each ventricle has its own choroid plexus that contributes more cerebrospinal fluid. Okay, so we've got some coming out of the third ventricle also. So from here, it's going to go down through the cerebral aqueduct, which is right here. Okay, that little narrow channel, into the fourth ventricle. And there's the choroid plexus of the fourth ventricle. Again, we'll add more to it. And then it will leave the ventricles through those apertures. So on this diagram, you can't really see the apertures, but they will lead into this space. You can kind of see where they're showing you one of them here. I believe that's the median aperture. So it will then enter the subarachnoid space, which is shown here in this purple color. And notice that will allow it to circulate all around the brain and all around the spinal cord. Okay, so as the cerebrospinal fluid travels through the subarachnoid space, it makes all these kind of curves, it's going to eventually go through the little arachnoid villi. Notice they are sticking up into this darker blue is the superior sagittal sinus. Okay, so it's going to go up through those, and will now be in the superior sagittal sinus. Okay? So it just mixes with the blood in the superior sagittal sinus, just becomes part of the blood plasma again. So it comes from the blood in the choroid plexuses of each ventricle, circulates through the ventricles, through the subarachnoid space, and then it will eventually go through the arachnoid villi into the superior sagittal sinus. Okay? So that's what they actually are, this whole thing here, is that's what it's saying. But for some reason, they started in the third ventricle. I'm not sure why. So I think you should add another step in here and start with the lateral ventricles. And remember, from here it goes through those little interventricular foramina. And these lead into the third ventricle. So this is really the starting point. Okay. And this where it says it moves into the venous sinuses, this really, I think it should say superior sagittal sinus. It's a little more specific. Okay? So there you have it.
I'm so sorry if you already told us this. What is the function of the cerebrospinal fluid?
Cerebrospinal fluid actually has many functions. It helps to maintain normal electrolyte balance in the nervous system, the central nervous system, so it helps to control the ion levels in the interstitial fluid around the neurons, and that's important for the conduction of action potentials. It also removes waste, which is a huge function. You guys, if I can hear you, everyone else can too. If you're not gonna listen, there is no reason for you to be here. Honestly, why are you here? Okay? And it also is a fluid cushion. That fluids the brain, the fluid cushions the brain and the cord from trauma, which is a huge function, really important. Okay, so other questions on any of that? Okay guys, so let's stop for now, let's take a break, let's come back at 8:45, and we will talk a little bit more about some parts of the brain, and we'll finish it on Monday.